Folks, I'm about to do another lecture on ecology, and let's see where I left off last time. So, through all this business on environmental factors. Now I'm going to pick it up here. I believe this is where we need to go. We'll talk about ecological associations among microorganisms. And so they fall into two categories. This is pertinent because in ecology, we're talking about interactions between organisms and their surroundings. And the surroundings include other living things. So biotic factors in the environment are things that we have to respond to. So that would be other organisms of other kinds, or maybe our own kind. So sometimes members of the same population that we belong to pose some issues for us. Either we cooperate with them or we compete with them. But in all cases, when we interact with other organisms, we're interacting with the biotic factors in the environment. Okay, and so those can be symbiotic or non-symbiotic kinds of relationships. So the word symbiotic comes from the word symbiosis, which means living together, right? So organisms that live in close relationship with one another are interacting in a symbiotic way. And they fall into these different categories. We've got mutualism, commensalism, and parasitism. I think I've talked about before how we can represent these with symbols. So in mutualism, two organisms live in close contact with each other, and they do better together than they would otherwise. So often these are obligatory. That means if one organism is found, you're always going to find the other one with it, living on or in it. So that's a mutualism. Commensalism is where one organism benefits, but the other one is indifferent, so it's neutral. It doesn't really gain or lose. This is like a hitchhiker. So if you've got an organism that lives on your body or in your body, and it's not really harming or helping you, but it in turn is gaining resources and it's doing well, that's commensalism. And then parasitism, this is the one that, most of us think about when we think about microbes. We think about the ones that make you sick, which in the grand scheme of things, is actually not as common as some of these other relationships. But they're the ones that are important to us because they disrupt our lives sometimes, as we're finding out right now. So if you've got a parasite, it lives on you and it gains, right? It's getting nutrition from you and a place to live, and you in turn are going to be harmed. Maybe not killed outright. You're not going to feel good. And in parasitism, generally that's the case. They tend to take just enough so that they can survive and reproduce, but they're not going to kill you outright. It would be, not beneficial to do that. And generally, parasites do evolve in that direction. They start off sometimes as being really serious in terms of their effects, but those over time become less and less and less because natural selection would favor that. Those organisms would, gain more from not harming you extensively than they would killing you. If they kill you, that's it. Their source of nutrition is gone. And then non-symbiotic relationships, these are where two organisms come into contact, maybe only infrequently, but they don't have to live together. It's something that's only temporary. It's only something that is for a short period of time, typically. So synergism is cooperation, where the members of the pair or sometimes larger groups work in such a way that they do better together than they would otherwise. but it's not a long-term, well-established connection that they have. And likewise, we have antagonism. Now, antagonistic relationships are also referred to as competition, and um, usually are symbolized with two minuses. So a lose-lose. So the thing that's key here with antagonism is that there's typically a limited resource that they're vying for, that they're competing for. Something that the interacting forms would all need, but it's in limited supply. So it could be space, or it could be some nutrient. It could be access to a host, whatever. That would ultimately be a situation where we would expect this kind of thing to happen. So those types of relationships are sometimes investigated by population ecologists. So we got symbiotic relationships. These are close, usually very intimate. We have the non-symbiotic ones, which are not intimate. These are instances where the relationships are, only temporary in many cases, and they're certainly not required for survival. And in fact, antagonism is counter to survival for all of the competing species. And so one example of antagonism, by the way, or competition, is antibiosis, and that is the root from which the word antibiotic comes. So organisms that are, oh gosh, that's horrible, sorry. The word antibiotic comes from antibiosis. An antibiotic is a chemical, and it's released by organisms that are involved in this kind of interaction. So they release them into the surroundings, and they seep into the soil or the water, wherever they happen to live. And the organism that produces it gains the advantage of keeping other potential competitors at bay. Right? So it's a kind of warfare. It's chemical warfare. That's what antibiosis is. Also sometimes called allelopathy. That's usually how I've heard it referred to. It's allelopathy. So the antibiotic chemicals that we use come from organisms that are practicing antibiosis. So penicillins, cephalosporins, erythromycins, chloramphenicol, on and on. Any of those natural products of fermentation that fungi and bacteria produce in competition, those are antibiotics. So anyway, there's that. And we mustn't forget that, the organisms that live on and in us are some of them commensal. Some of them are there as synergists. They're helping us, and we're helping them in turn. I guess if it gets to the point where it's obligatory, and there may be some of those, we would call it mutualism. We have hundreds, maybe thousands of species in our bodies, in our guts, on our skin, up our nose, various other compartments of the body, and they make up this microbial flora, which is basically just a big community of organisms, including bacteria, fungi, protozoa, and even archaea. They should be listed here as well. Now, sometimes, this is not something we want to happen, obviously. Sometimes those members of the flora can turn on you. Sometimes they become too abundant and start to take over and make us sick. So infections, unfortunately, happen. Organisms that do this, more often than not, are not strict pathogens, but they're opportunistic. So they're just exploiting, immune um, you know, an immune response that's diminished because of stress or some other factor. Maybe we're enduring chemotherapy or something like that. So um, that doesn't unfortunately happen sometimes. But yeah, this normal flora, and we're learning more about that. Been in the last 10 or 20 years that people have really begun to appreciate just how important it is. These are organisms that are there and should be there. If they're not, then that would signify major problems. And then there are biofilms. A biofilm is simply a population, or maybe several populations forming a community. You might have one species or multiple species occurring in a biofilm, but organisms that are, capsule formers, produce a glycocalyx that's really thick, can form these biofilms. And we're finding out more about these as well. We find that in some environments, these are dominant. These are really important. Like, say, for example, the bed of a stream. If you were to go to a stream and examine the surfaces of the rocks there, often you'll find an incrustation of these organisms. That community is referred to as periphyton. So algae and bacteria and other kinds of things encrusting the rocks, making them slippery in some cases. That's periphyton, and that's a form of biofilm. It's a biofilm that occurs in a stream type of environment. And they occur elsewhere. In our mouths, we have a biofilm on our teeth. We try to scrub it away every day by brushing our teeth, but it always comes back. There's a biofilm of organisms occurring on the surface of the mucosa in our gut. So, we've got this kind of thing happening in lots of cases. There's this interesting phenomenon that occurs in biofilms as they develop. There's this form sensing, which is a kind of communication using inducer chemicals. The members of the community, the biofilm community, release these, and they pass from one organism to the next, and they actually cause DNA to certain genes in the DNA to be expressed once the population gets large enough, once the biofilm becomes extensive enough. So this shows a compartment, say, in a person's gut. And initially it's like this, there's just, a few organisms there. But as they settle, they begin to divide and they become more numerous. And they start secreting these inducer chemicals. And those are passed from one individual to the next. And this shows what happens when they're taken up. They bind to the DNA in the recipients. And it's a chemical cue that says, hey, start expressing this gene. And so the gene that's activated is one that produces a protein, which can be released into the surroundings and serves as an exoenzyme. These are enzymes that function outside of the organism's body, and they help with the digestion of food particles. So this is in somebody's gut. There's food coming through there. Big particles get broken down into smaller and smaller pieces, and those in turn can be taken up by the organisms, and maybe even the host that is, being used as the substrate for the development of this biofilm. It could make use of those substances. But we get more nutrition this way than we would if we didn't have the biofilm. That might be one possible outcome of that. So yeah, biofilms are typically composed of organisms that have this capacity to form capsules. And it's that capsule material that polysaccharide. You see all this sort of hazy looking stuff. That's the glue that holds the biofilm together. And then form sensing is something we see in. All right, and so we want to talk about the study of microbial growth, population size increase. Ecologists are interested in this because organism function, how well they take up nutrients and the interactions they have with one another, can influence the rate at which population sizes grow. And it's also a function of genetics. Different species have population growth that occurs at different rates. Now, the baseline type of growth that we see results in a growth curve. We call this a growth curve, where the x-axis, this horizontal axis, is time, and population size starts off small, but over time, it's bigger and bigger and bigger, and it takes off in a fashion that leads to the formation of this J-shaped sort of curve. It's got this sort of bend to it. It doesn't go straight up like this with a uniform slope, but it starts off with a low slope and it becomes steeper and steeper as we go up. That's called exponential growth. Or some people call it geometric growth. All right. So it's growth that follows an exponential mathematical sort of function. And what's happening in each time unit, right? If you were to divide this up into different time units, in each unit, the amount that's added to the population gets bigger and bigger and bigger and bigger. What happens is this: you start off with a single cell because bacteria are asexual. You can start off with a single cell, and that one cell can divide. Right? That's a single cell. It divides into two, and those two divide into four, and then 4 divided to 8, and 8 divides to give us 16, and so on and so forth. So there's this doubling at these intervals of time. And that varies from species to species. How long does it take for the doubling to occur? Some it's only just a few minutes, and some it's, say, big chunks of an hour. In some cases, it's hours or days where populations double. So different species grow at different rates. But all of them initially are going to exhibit this exponential sort of J-shaped growth. Later, of course, that's going to change. I mean, unless the environment is infinite, which in most cases it isn't, they're going to start to run out of resources, and then the growth pattern is going to shift. Yeah, binary fission. This is where organisms that are asexual literally just duplicate their chromosome and split in two. Yes. So that's binary fission. We're talking about asexual organisms, bacteria and archaea and many protista. This is how they grow. Not by mitosis. Mitosis is the type of cell division we see in eukaryotes, but binary fission. So that's a pretty complicated process. We're not going to get into that in great depth. We'll just go ahead and leave it there. But this is what you get, right? So this model over here on this side shows what I was just showing, what the actual number of cells would look like at each interval. And you can see after 5 generations of growth, there's 2 to the 5th power, or 32 cells. After 6 generations, there'd be 2 to the 6th, 64. 2 to the 7th, there'd be 108. It just goes up like that if we follow this sort of exponential model strictly. Some species don't follow it that closely, but at any rate, I mean, you're going to wind up with a J-shaped growth curve. Now you can straighten that curve out. Sometimes it's efficacious to do that. Sometimes it's helpful to linearize this, and you can do that by plotting these exponents. You take these numbers and figure out what number applied to the base of 2. So what number n applies to the base applied to the base of 2 would give you each of these values. So like 2 to the 9th power is right around, it would be the exponent, 9 would be the exponent. When applied to the base of 2 would give you 500. 2 to the 10th would be right around 1,000, and so on. So if you plot these values instead of the whole number values, you're going to linearize your curve. It's going to take the bend out of it, the J out of it. Sometimes that's helpful to do that. That's exponential growth. And that's what typifies all populations in the beginning. All populations, whether they're sexual or asexual, have this capacity. Do any of them do it forever? Nope. Eventually you run out of resources, right? And the population growth pattern would change. Let me show you what that looks like real quick. Let me plug in slide. So if you were to extend the population growth investigation out for longer for a given species, and N, by the way, is the symbol for population size. Initially, populations are small. There's lots of resources, and individuals double. They divide, and you have more organisms coming into the population than you have dying. So the curve is always going to look like this. Initially, it's going to be exponential. But if it could keep going indefinitely, it'd just take off like a rocket. Throughout all time, it would keep going up. But do they ever do that indefinitely? No. At some point, there's going to be a leveling off. Why? Well, the population gets dense. There's lots of crowding. There's wastes being produced. That sort of fouls the environment. Resources get taken up, so there's not as much nutrition. Things get bad, and so the population starts to slow down. And it's still expanding, but at a slower and slower rate. It's not doubling anymore every generation. It's doing something less than that. And eventually you flatten out entirely. And the population will reach this sort of plateau. And that's called the carrying capacity for that particular environment. That's the carrying capacity. And some populations may go on and on like that for a long time. And you can see now that it's no longer J-shaped, but it's more like an S, right? So that's called sigmoidal growth, or logistic growth. Gets to looking like that. That indicates there's some limitation going on. At that point, the number of births is exactly equal to the number of deaths. Down here, where you got exponential growth, birth greatly exceeds death. Now, what do you suppose is going to happen if we keep going? We follow this out and there's no new resources and the environment's not refreshed. It's actually going to start to tail off. You could have enough time to follow this, and it may actually go down to 0. What's happening here? Well, births are less than death. Death is predominant further on in time you go. Unless something happens to make that environment better, to enrich it, that's typically what will happen. So anyway, one of the things that people who study population growth in different kinds of organisms do is they compare these species with regard to the, the conditions under which they grow best. And that's helpful for a lot of reasons. I mean, it can help you to determine if you're trying to grow an organism on purpose. It can help you determine what the best conditions are. If you're trying to kill the organism, if it's an infectious organism, you can figure out about when to expect its population to decline so that people can go back to normal, as we're doing now with the coronavirus. Studies like these are very informative. So, yes, that's exponential growth is J-shaped growth. It ultimately becomes S-shaped if we follow it for long enough. And finally, you can reach a point where populations decline if the environment's not. One way to estimate the size of a population, at least initially, that seems to work pretty well, is to use a mathematical model like this. So this is the exponential growth equation. Exponential model. An equation like this can be referred to as a model because it tells us what to expect in the future in certain limits if we apply this. And this one is specific for asexual organisms like bacteria. A slightly different form of this is used to estimate population sizes in the future for sexual organisms. But here, N sub I, this term right here is the starting population size. So if you were starting with one cell and you wanted to say, figure out, how many cells will there be 10 days from now or 6 hours from now, 1 would be the number you'd put in here. Or it could be any starting size. You could have a population of 1 or 10 or 100, doesn't matter. But that would, whatever you're starting with, that's N sub I. And then where you're winding up, that's the number of individuals you expect, you're predicting, not the actual number that would exist, like the prediction of where the population size would be after some period of time has elapsed. Final population. And the number of generations into the future that you're projecting as N. Says N denotes the generation time. Well, it's actually the number of generations into the future, which is different than generation time. Generation time is the length of time that's elapsed in minutes or hours or days or whatever the appropriate time unit is. Number of time units that's elapsed during which the population has expanded, has increased, has doubled. N is the number of those time units into the future we're projecting. So let's say the generation time for organism X is 15 minutes. That is, if 15 minutes elapses and you have a certain population size, say 10 that you're starting with, 15 minutes, it'll double to 20. Then another 15 minutes on, it'll double to 40 and so on. It's going to expand every 15 minutes. That's the generation time. Another species, Y, might have a generation time of 30 minutes. So if you start off with 10, then 30 minutes later, it's going to go to 20. An hour later, it's going to go to 40. So that one's growing slower. So N here is the number of those generation times that have elapsed over the time into the future that you're projecting. So let's say N sub I is 2. And let's say the generation time is 15 minutes. We want to know what N sub F is after 2 hours. What is N sub F after 2 hours. Well, the generation time is 15 minutes. How many 15-minute periods are there in 2 hours? Well, there's 4 15-minute periods in an hour. An hour, 60 minutes divided by 15 is 4. So in 2 hours, there's 8 generations that will have elapsed. Right? 2 hours, there's 8 generations that will have elapsed. So n, this number here, will be 8. Right? So n sub f, using these numbers, 2 hours, is 2 times 2 raised to the 8th power. I think that's 6. It's 512, I believe. I believe. I did that off the top of my head. I think I'm right. I'm not for sure on that, but I think anyway, that's how you would use that. And so some other species, let's say the generation time is not 15 minutes, let's say it's shorter than that. After 2 hours, you would expect more than 512. Or if the generation time is longer than 15 minutes, let's say it's a half an hour, you're going to expect fewer organisms to arrive. So yeah, that's the exponential model. And like I say, at least initially, and for some time thereafter, populations will conform to that. But eventually they're going to run out of resources, and you'd have to have a different model, one that follows the sigmoidal growth curve. Anyway, so how do they figure out generation times and all of that for different species? Well, one way to do that is to perform what's called a viable plate count. So viable means alive, and plate refers to an agar plate, right? And so here's one way that you can do this. So this represents a flask of molten agar that's been cooked, it's liquid, and they place a small number of organisms in right at the beginning, right, just to inoculate it. And if you were to swirl that up and then draw out of it 1/10 of a mil, which is enough to make an agar plate, pour that out, right, and place it in an incubator, and let it solidify and let it sit. You probably won't see any growth. You won't see any colonies because the number of cells to start off is so small, the chances of any of them being there would be almost zero. And so that would, you would just by default say, well, there's cells there. I know they're there. I inoculated it. I know they're in there, but the growth hadn't gotten to where it was substantial. I'm going to say that the number of cells initially was less than 5,000. And by the way, this is after having incubated 60 minutes. So it's incubated, but not long enough apparently to organism. Now, this is the same after 120 minutes. That same liquid agar is sampled. We're keeping this agar, by the way, in a bath that keeps it at the right temperature to allow it to stay liquid. We draw off another 1/10 of a mil. We make another plate, and this is after 120 minutes. We let that incubate, and we see there's 2 cells in there. 2 colonies, indicating 2 CFUs. There's 2 per 1/10 of an ml. You had 500 mLs to start with. So if you extrapolate to how many cells total you would expect, there's 10,000 in that flask at that point. And then 3 hours later, you plate them out, you get 4 CFUs. That translates to 20,000 total cells, right? and then it just goes up. But if you were to take these numbers and graph them, right, you could see, you know, what the shape of that curve is, and I'll just tell you, it's going to appear J-shaped, right? And so you can estimate the generation time from this. You can maybe figure out about when the population is going to transition into a sigmoidal kind of growth. You can learn a lot about a particular species and its growth pattern using an approach like this. If you did the same study with a different organism, say this is E. coli here, did it with Staph aureus or some other thing like Salmonella, you might see a totally different pattern. Okay, that's called viable plate count. You're putting organisms into a medium, you're sampling that serially, and you're placing them into agar plates and then looking to see how many CFUs you get. So if you were to do a study like that and extend it for long enough, you might see the population going through these different stages. And what they've plotted here is not the actual numbers of the population size at each point. They're not plotting these numbers, but they're plotting the exponents that you would apply to the base of 2 to get those numbers. So, 2 to nth power is, and you plot that. 2 to the nth power, will give you 675,000. You figure out what n is and you plot that. That's what this is showing. So these are logarithms. These are exponents applied to the base of 2. When we plot those, it straightens our curves out and makes it nicer. See, initially, you've got a slope that's shallow and it's a straight line, but here it starts to take off in an upward direction of a straight line. That's the exponential phase. And they've done this study for long enough that you see there's a stationary phase where it flattens out. In the stationary phase, births equal the number of deaths. In the death phase, births are less than deaths. And this could go down to 0 at some point. All right. So again, you could perform studies like viable plate counts for different species and graph the results this way and compare species, see whether there's differences among them. By the way, in these tubes, you can see how it starts off real light, and then it gets a little darker, and then it gets a little darker. That darkness is turbidity. That's cloudiness of the culture. You can see it gets darker and darker. It's becoming more turbid. And in the bottoms of these, you can see some sediment forming. Those are dead cells. So we're recording death here as well. So in addition to viable plate count methods, other ways of analyzing population growth would be to take a tube, inoculate it. So there's a newly inoculated tube, and at first it's going to be totally clear. If you let it sit for a while, it's going to start to get turbid, right? And if you let it go for an extended period of time, it's going to get more and more turbid until death starts happening, and it's going to start to become clear again. And you're going to see sediment building up in the bottom. But there's ways to actually measure that degree of turbidity, and that's called turbidometry, and it uses this machine called a spectrophotometer. This is another way. You don't have to even make auger plates. You have a well-functioning spectrophotometer. You can just use that to pass light through your sample. And the extent to which the light is absorbed is going to give you an idea about what the population size is. So this first tube, this one that's clear, if you put that in this machine passes light through it, and there's a reader that says, how much light got through the sample. Here's our sample. It's totally clear. These are the photons of light. They're going straight through. And the reading here is pretty high. That absorbance reading is one for the nutrient broth. which has some solute in it, so there's going to be some absorbance. But it's a reading we would get if there were no growth at all. So that represents zero growth. Sometime later, we'll take a more turbid tube, and you can see the amount of light getting through is starting to become less. These are broken lines, so it's less. The absorbance is higher. Let's say this is after 15 minutes. Probably longer time period. But if you were to take that tube, that same tube, and measure the absorbance at 15-minute intervals, you'll see it becoming more and more cloudy over time. And you record those absorbances. You could do that for different species and compare them that way. So this is not directly measuring population size, but it gives us relative population size. And it's quantifiable. I mean, we're actually recording things, but we're recording absorbances instead of actual numbers of cells. But that's a way to go. And then finally, this is not final. This is another way we can estimate population size. We can do a direct cell count. A viable plate count was what I showed earlier, where you're counting CFUs and using that to determine how many cells were in your inoculated flask of broth or agar. Here we're actually taking a pipette. This is a pipette tip. We've drawn into it a known volume of some growth medium. Let's say it's 1/100th of an ml of a small amount of some sort of broth that has organisms in it. Put them on there into this little grid system. And each little square represents a particular amount of area, right? So you can count the number of cells per square in that system. You know that whole thing represents 1/100th of an ml. You can then go ahead and extrapolate from that back to what the original volume had in your 1/100th of an ml, and you've got however many cells are here, 13, let's say. Let's count 1, 2, 3, 4, 5, 6, 7, 8, 12, 13, 14, 15, 16, 17, 18. 18 cells for 1/100th of an ml. Just plugging in some hypothetical numbers. It would take 100 mLs to equal 1 ml, so 18 times 100 equals the number of cells in 1 ml, 1, and say 1,800. In a whole liter, there's going to be 1,000 times that many. So 1,800 times 1,000 would equal the number of cells per liter. So that's 1.8 million. Million is 10 to the 6th power. You don't need to know that. That's the number of cells per liter. That's how you could do a direct cell count. Started off with that known volume on that little grid system. You count the number of cells, and then you work your way back to the original total volume. Estimate the number in there that way. And then you can use a direct cell count method that's automated. Okay, so this one back here is manual. Using this system as manual. This is an automated system. They use machines called flow cytometers. I've never used one, but I had a friend who actually sold them. They can count blood cells. They can count different cells using these. And this, they have them set up to count bacteria. You put your sample in this machine, and it's got a little laser light in it. And the sample is passing this very tiny little portal, which is wide enough for a single cell to go through at once. So it takes a while for this to work, but cells go through, they trip that laser light. It turns out that dead cells and live cells produce different results. The amount of laser light that gets through the cell is variable depending on whether they're living or dead. So not only can you count individual cells directly, you're not figuring out relative numbers, you're actually counting numbers of cells. You can not only do that, but you can tell whether they're alive or dead. So that's really useful. All right. So you can compare organisms that way. So this plot here is for Staph aureus, and it shows after however much time had elapsed in the growth of this broth, growth of this culture. There were, in that range, say 10 to the third power is 1,000. There were 1,000 or so within limits. There's a whole range from here to here of cells that were living, and then this number dead cells. E. coli, you can see after that same amount of time had lots more dead cells. The green represents dead cells. Those conditions that favor Staph aureus apparently don't favor the growth. But that's using a flow cytometer to directly count cells. Automated method. All right, so there's that. I'm going to go ahead and stop there. Go back and see. I think there might be a chunk of this PowerPoint that I need to lecture on. I may have skipped over a part. I'll have to check and see. But if not, I'll let you know. And I'll be posting an exam on this stuff shortly, like either tonight or tomorrow. And I'm going to have the due date for this one on the 23rd. So your virus test is up there now, and the due date on that is the 16th. So sometime between now and then, if you haven't already done that, you need to do that. This one I'm going to make available soon, and I'll have, whenever it goes up, when it's due, it'll be a week. So I'm guessing that's the 23rd or the 24th. All right, so I'm going to stop there and just forge ahead on. I know this is not ideal, and I'm struggling because I got all kinds of stuff going on at home. I'm having to do this from home. I've got a house full of people coming and going, and it's a challenge. So apologize for lack of timely service. It's a pandemic, so think about that. Before it's all done, things could get a whole lot worse than they are. We don't know where we're headed. So anyway, let's just stay healthy, social distance, wash your hands, take that seriously. A lot of folks aren't taking it seriously. I, for one, am taking it very seriously. A lot of people aren't. It's a shame. It could spell disaster for some folks. All right, so I'm going to go ahead and stop right there, and I'll be posting more stuff shortly.